Hello, today we are going to talk about protein synthesis. My name is Jimena Lopez and my team member is Daniela Galvan, which will appear in the next explanation. Protein synthesis occurs during transcription of DNA into RNA and then the translation of it into proteins, which are made of amino acids. The instructions to make proteins are in our DNA. Each gene in the DNA, which the DNA is compacted in the chromosomes, and it has in every DNA there are like instructions for each type of protein in our body.
Well, replication occurs during mitosis, and replication is the cell division. In here, as you can see, we have the parent DNA that is the helicase, and then this double helicase divides into the two parent strands that serve as template, and then the DNA polymerase comes and starts creating two new complementary strands that are here with 4 different nucleotides that are adenine, guanine, cytosine, and thymine. Adenine always goes with thymine, and cytosine always goes with guanine. And the two new complementary strands that the DNA polymerase created are the daughter strands that are complementary to these parent strands and have the same DNA.
Now I'm going to talk about the first process of protein synthesis, transcription. Transcription occurs in the nucleus. And in transcription, DNA is used as a template to make RNA. One important thing to take into consideration is that during this process, during the arrangement of complementary bases, instead of using thymine pair with adenine as normal replication, adenine pairs with pure uracil. So therefore, RNA never has thymine. Now I will explain the process. First, the enzyme RNA polymerase, this blue thing, comes into action and unwinds the DNA helicase, unwinds it, and then starts matching the complementary, the ribonucleic complementary nucleotides. As I said, instead of thymine pairing with adenine, uracil is paired with adenine, and therefore it starts making the RNA strand. It's only when the RNA polymerase reaches the end of the gene, the RNA strand is completed. Then, when the RNA strand leaves, it goes through the nuclear pore into the next step. Translation. During this process, which is a final process of protein synthesis, the mRNA is transformed into amino acids. After the mRNA leaves the nucleus, it enters the cytoplasm. The mRNA has codons which are groups of 3 nucleotides, as you can see here. Then specific tRNA come with anticodons, which are complementary to the mRNA codons. As you can see here, this is a tRNA which has a specific amino acid, and it has an anticodon which is, as I said, complementary to the mRNA codon. This happens in the ribosome. Yes. Each type of codon translates into a specific amino acid. There are 64 different possible combinations of codons. As you can see in the chart, these are each combination of the three nucleotides, which are codons. For example, here, UAC translates into a TYR amino acid. There is one codon for start, a specific codon for start, and three specific codons for stop. And here we can see a GIF of the process. Here comes the tRNA, which is complementary to mRNA with the specific amino acid, and then leaves, and a chain of amino acids starts forming. And when it finally reaches the stop codon, the process stops and the amino acid is produced. As you can see, this is the final chain of amino acid, which then produces a complex structure of a protein. This is a summary of these three stages that we just saw. Replication is the process during cell division or mitosis when two complementary DNA strands are formed from one parent DNA. Transcription. Transcription is the process by which DNA is transcribed into RNA with a ribonucleic polymerase, which transcribes the DNA. Translation is the process by which mRNA is translated into specific amino acids which have a certain function.